know about beta thalassemia?
No! What is that?
Oh, I know. Beta thalassemia is a type of genetics disorder.
That is correct. Let's dive more
Into it!
Genes are the building components of inheritance. They are passed down from parents to children. They include DNA, which give the instruction for producing proteins. Proteins are responsible in a majority of the work in cells. They transport molecules in between body location, construct structures, degrade poison, and perform numerous other maintenance tasks. A mutation is a change in one or more genes. Some mutation alters the gene's instruction for protein production, resulting in a protein that does not function properly or is completely missing. This can result in a medical disease known as a genetic disorder. There are three types of genetic disorders. The first one is single gene disorder, where a mutation affects one gene. The second one is the chromosomal disorder, where chromosome or parts of chromosome are missing or changed. Chromosomes are the structures that hold our genes. And the last one is the complex disorder, where there are mutations in two or more genes. Often your lifestyle and environment also play a role. Now I will pass this presentation to Casper to talk a lot more about our topic.
Good morning everyone. I'm going to explain what is beta thalassemia, which is a blood disorder that was treated using caspase-9 CRISPR gene editing in 2019. So, what is beta thalassemia? Beta thalassemia is considered as a form of anemia, which is a condition characterized by a deficiency in the number or quality of red blood cells, which impairs the ability of the blood to carry electrical oxygen to the body's tissue. So, what kind of mutations in beta thalassemia will cause the genetic disorder? Let me ask you what is happening in beta thalassemia patients. Beta thalassemia was caused by over 300 mutations in the adult beta globin gene. The symptoms are highly prevalent in underdeveloped nations due to the genetics counseling of lacking and the prenatal diagnosis. If we look at the genetics point of view, the mutation in genes in beta thalassemia occurs that reduce or eliminate the beta globin expressions. Also, it will cause the increased number of alpha globin formation because the formation of the heterotetramer of hemoglobin is not complete. Next, the excess alpha globin change form the insoluble aggregates, leading to the ineffective erythropoiesis and apoptosis in the red blood cells. The imbalance between the alpha globin and beta globin chains cause the oxidative damage to the red blood cell precursor, further develop the beta thalassemia disease.
Beta thalassemia is an inherited blood disorder characterized by low levels of functional hemoglobin, which is the oxygen-carrying pigment in our red blood cells. It comes in three main forms: minor, intermediate, and major, indicating the severity of the disorder. Minor usually shows no symptoms and thus is called as asymptomatic, while individuals with beta thalassemia major, they have severe expression of the disorder and requires regular blood transfusion and ongoing medical care. Intermediate thalassemia, on the other hand, is less severe than thalassemia major, but much more severe than thalassemia minor. They may experience symptoms such as fatigue, weakness, pale or yellowish skin, and enlarged spleen. The severity of the symptoms can vary widely.
Now we will look into the clinical application of gene therapy in treating beta thalassemia. First is the in vitro experimental model systems erythroid precursor cells from beta thalassemia patients. When developing gene therapies for beta thalassemia, testing the safety and effectiveness is crucial. Hematopoietic stem and progenitor cells (HSPCs), which can develop into red blood cells, are used for this test. These cells are often taken from peripheral blood because they are easier to access than bone marrow or mobilized blood, which are preferred for actual treatments but more challenging to obtain. The culture process is a two-step procedure. Initially, the cells are cultured in the presence of combination of growth factors, excluding a hormone called erythropoietin or EPO, which stimulates the production of red blood cells. This phase is designed to promote cell division and multiplication, thereby creating a larger population of precursor cells. After that, EPO is introduced, aiding the cells in maturing into red blood cells.
The next clinical application of gene therapy is globin gene addition technique, specifically through lentiviral expression of exogenous beta globin. The field of hematopoietic stem and progenitor cells, HSPC gene therapy, aims to achieve efficient and stable transduction of cells, optimize the expression of the introduced transgene, and correct beta thalassemia in preclinical models while minimizing genotoxicity. Enhancements that can be done for better outcome is number 1, rapamycin, which has been shown to enhance lentiviral vector transduction, making the process more efficient. Second is chromatin opening elements and N-chirin insulator, which are used to improve the expression of the transgene delivered by the vector. They ensure that the vector-derived beta globin is expressed at higher levels. Third is alternative insulators, which these elements prevent the silencing of the transgene and minimize disruptions to host genes, ensuring a safer and more stable integration.
Gene editing in beta thalassemia using CRISPR includes one of the most exciting aspects of CRISPR-Cas9, which is precision targeting. This technology allows scientists specifically edit the genes associated with beta thalassemia, offering a tailored approach to the treatment. Next, in 2019, researchers explored a fascinating strategy. It is to reactivate the production of fetal hemoglobin HbF using CRISPR-Cas9. Fetal hemoglobin can compensate for the defective adult hemoglobin in beta thalassemia patients, potentially reducing the symptoms and improving quality of life. Sadly, preclinical studies in animals showed promising results. Therefore, by correcting gene mutation with CRISPR-Cas9, researchers observed increased production in fetal hemoglobin and improved in disease symptoms. In addition to preclinical research, early-stage clinical trials were initiated to evaluate the safety and efficiency of CRISPR-Cas9-based therapies for beta thalassemia. Now, let's dive more into HbF activation. HbF activation is done by genome editing. It is to compensate for the defective adult hemoglobin in beta thalassemia patients. By using CRISPR-Cas9 to edit the genome, researchers aim to awaken the production of fetal hemoglobin, which normally will decrease after the birth, but it can still be produced throughout life, to make the symptom of beta thalassemia better.
And now me and Valerrie will explain about the symptom of the beta thalassemia. Different people will have the different symptoms based on which type of the disorder is inherited. The first is the beta thalassemia minor, also known as the beta thalassemia carrier or beta thalassemia trap. This is the mutation in the HBB gene which code for the beta globin change of the hemoglobin. Patients with this beta thalassemia minor usually have a mild anemia, which is the microcytosis, where the red blood cell is smaller than the normal, and hypochromasia, where the red blood cell has less color than the normal red blood cell. This might include mild fatigue and slight weakness. Next is the beta thalassemia major. This is the most severe type of this disorder. It also known as a Colles anemia. Children who are born with this type of beta thalassemia will have symptoms in early life. Individual with beta thalassemia major inherited two mutated HBB genes. This type of beta thalassemia will leading to pale skin, bone deformities, particularly in the face and skull due to the marrow expansion, jaundice, which is the yellowing of the skin and eyes due to the hemolysis, which is the destruction of the red blood cell. And last is the abdominal belly.
Beta thalassemia intermediate is a milder form of disorder which is less clinically severe compared to beta thalassemia major, not as severe to require regular transfusion. Symptoms of beta thalassemia intermediate are: number 1, fatigue, an extreme sense of tiredness and lack of energy that can interfere with a person's usual daily activities. A person with fatigue may feel weak, worn out, or run down. They may also have trouble speaking or concentrating, or short-term memory loss and mood or emotional changes. Second one is pallor, condition of being pale. Third one is gallbladder disease due to gallstone formation. Number 4 is spleen enlargement, occurs due to erythroid hyperplasia, whereby massive productions of red blood cell in response to peripheral red blood cell loss or destructions or ineffective erythropoiesis. Intermediate and major beta thalassemia are both homozygous mutations that causes more severe spectrum of anemia. These two are distinguished clinically by transfusion dependence. The DNA testing that helps determine thalassemia cannot reliably predict whether a child is a major or intermediate. That determination is dependent upon the transfusion needs of the individual. Therefore, beta thalassemia major requires more routine transfusions, meanwhile beta thalassemia intermediate does not need.
Now you know. Thank you.
Sini.
Okay, Christy.